Good morning, distinguished guests, ladies and gentlemen, viewers online worldwide. Welcome to the 2022 Tom Price Masters Forum in Biopharmaceutical Science. This forum is organized by the Tom Price Foundation in collaboration with National Tsinghua University. Today's topic is lipid nanoparticle, gene therapy, and COVID-19 mRNA vaccines. We are honored to have the 2022 Tom Price laureate, Dr. Peter Collis. He will join us remotely from Vancouver. Before the forum starts, we would like to take a group photo. Please remain your seat. Our photographer will take a photo from the stage. Everyone, please turn to the stage. During this forum, our moderator will be accepting questions through slido.com. Please scan the QR code or click on the Slido link below the live stream channel. Thank you for your cooperation. Without further ado, we would like to invite to the stage the president of National Tsinghua University and also the moderator of today's forum, Dr. Gao Weiyuan, to deliver opening remarks and introduce our laureates. Ladies and gentlemen, please join us in welcoming President Gao.
Good morning to the people in Taiwan, and good evening to the audience joining us from North America and elsewhere. Thank you for joining us this morning. My name is Dr. Zhang Gao. I'm the president of the National Tsinghua University here in the lovely island of Taiwan. As we know, the year 2022 has been a very significant year. The world is emerging out of the shadow of COVID-19. It's also a very significant year because NTHU, we have launched our medical program and started our teaching hospital project with a commitment to educate future research scientists and clinicians with a passion to serve underserved communities. And the hospital is also positioned to facilitate the translation of innovative technologies to address unmet clinical needs and the world-class clinical service to the people in Taoyuan and around Taiwan. Consistent with the NTHU core values of inclusivity, equality, and diversity, medicine is a profound direction for the future growth of NTHU. This is also consistent with our new strategic plan, where we're putting people first. This echoes the mission of the Tom Prize, known as the Asia Nobel Prize. The prize was established to promote significant contributions to humanity. The Tom Prize in Biopharmaceutical Science recognizes work in biomedical and pharmaceutical research. I myself also have a pharmaceutical background, so it's delightful to meet Dr. Collis virtually through this program. His work has a clear impact on the prevention, diagnosis, and treatment along the patient journey for numerous diseases. As we know, during the dark time of COVID-19, vaccine development was an urgent global matter. Thanks to the thousands of dedicated researchers and scientists worldwide, today we have many options to choose from. One of this year's winner of the Tom Prize in Biopharmaceutical Science, Dr. Peter Collis, has pioneered lipid nanoparticles, LNP, to encapsulate nucleic acid-based drugs and effectively deliver them to the intended targets. This groundbreaking technology has led to the successful development of mRNA vaccines and therapeutics. Allow me a few minutes to introduce Dr. Collis. Dr. Collis has published more than 400 papers and more than 80 patents. That itself is already very significant. He's also the officer of the Royal Society of Canada and a professor in the Department of Biochemistry and Molecular Biology at the University of British Columbia in the lovely city of Vancouver, Canada. Our LNP platform is instrumental to 5 approved drugs. As we know, drug formulation and delivery remain a technical challenge for many promising therapeutics. These efforts have led to 5, not only these drugs, but also the foundation and formation of his company, Nanotherapeutics, and he has many interactions with many startup companies and established companies in the industry. He's also the founder of Pan-Canada Research Centers, including the Center for Drug Research and Development and Nanomedicine Innovation Network. Needless to say, Dr. Koulis has received many, many prizes, including the VinVenture Grand Prize and the Canada Gardner International Award and Governor General's Innovation Award. Today, he will tell us more about the translational journey of the LNP system, including the application in COVID-19 vaccines that benefited so many of us worldwide. Let's welcome Dr. Koulis, and I look forward to learning much more. Thank you very much.
Thank you very much for that great introduction. I'm going to just get my my screen up here just a minute. Can everybody see that okay? I can't really hear very well. It's a bit garbled.
Yes. Yes, we can hear you.
Here we go. Okay, great. I just want to say, first of all, what a pity it is that I'm not actually there. I guess the COVID restrictions are still in force in Taiwan, and it seemed like so hopefully I'm going to get there next year. I'm really looking forward to visiting you in Taiwan. The second thing is, of course, what a remarkable honor it is to receive the 2022 Tang Prize. This is, I mean, it really is unbelievable, really, from my point of view, that we've received this kind of recognition. So what I'm going to talk to you about today, and I'm going to keep it at a fairly high level, but it concerns lipid nanoparticles, gene therapy, and the COVID-19 vaccines. But really, this is emphasizing my very long association with lipids, 50 years of lipids. I have a number of conflicts of interest, as is indicated here, from companies that I've co-founded. Now, you've probably seen the headlines such as this. The vaccine revolution is coming inside tiny bubbles of fat. This is a headline from Bloomberg. And this is the lipid coating that goes around the mRNA that is eventually injected into us and produces the immune response. That's, of course, vital as to the vaccines. So let's go back to this tiny bubbles of fat. What are these tiny bubbles of fat? They're already pretty tiny. They're 100 nanometers in diameter, so about 100 times smaller than a cell. But they're really not bubbles of fat. So they're nanoparticles, at least not fat as you normally think of it. They're nanoparticles made of membrane lipids, and they've really taken quite a long time to develop. And so what I'm going to talk about in part is work that I did from about the early 70s, and it's still going on now. Basic studies on membrane lipids. We still don't understand the great variety of functions that they serve. I won't talk too much about this. Between 1985 and 1995, we developed systems to deliver cancer drugs. But since 1995 to now, we've really been focused on developing systems to deliver nucleic acid-based drugs into cells in vivo. So just to begin with, and this is for students in the audience, that when you graduate with a PhD, it doesn't necessarily mean that you're going to have to continue doing that all your life. I got my PhD in physics using magnetic resonance. And after getting my PhD, I decided I really wanted to do something different. It seemed like the most interesting problems were outside the field of physics. And so I got interested in applying nuclear magnetic resonance in a life science context. And surprisingly, I was awarded a Medical Research Council postdoc to go to Oxford University in the UK. I really didn't know anything about biochemistry. I've taught biochemistry now for pretty much 40 years. I never have taken a biochemistry course. But it's as I say at the beginning, I knew a bit about nuclear magnetic resonance, NMR. And this is my last paper actually as a physicist. That 96 equations, I couldn't reproduce them now, I can tell you. But what do you learn as a physicist? You learn a technique, obviously, in some depth. You also learn that when you need an instrument that isn't available, you build it. And of course, the most important thing is you go after basic problems. When I got to Oxford in 1973, they had a magnet, but they didn't have all the electronics. This is in the background here that are required to actually do nuclear magnetic resonance. And so, with me and a graduate student, we had to actually build this stuff to get this NMR machine going. So that was kind of my physics background came in quite useful there. So going after basic problems, and what we did was we tuned up the NMR machine for phosphorus, so we could look at phospholipids, and then really just started to investigate certain basic problems. So I got interested in the properties of lipids in biological membranes. So this is a Singer-Nicholson model of a biological membrane, which really is still quite valid today. Lipid bilayer penetrated through by integral proteins and also partially by peripheral proteins on the outside or on the inside. Now, why are lipids interesting? They're not really, often not really emphasized in biochemistry courses, but I find them absolutely fascinating. So in your body, you have more than 30 trillion cells. And they're all surrounded by a membrane, whether they're in the muscle or pancreas or kidney or wherever they are. And so each cell is surrounded by a membrane. And then, of course, you have all the organelles inside a cell, and they're all surrounded by a membrane. And so these membranes, of course, separate the inside from the outside and allow very specialized functions to occur within the cell and, of course, within the organelles themselves. So all of these membranes rely on a lipid bilayer to provide this permeability barrier. And the particular type of lipids that make up this lipid bilayer, they're membrane lipids. They're not fats, of course. They're amphipathic. So one end likes water, the other end is hydrophobic. And so they're pretty vital to life. If we didn't have lipids, we wouldn't exist, that's for sure. I spent nearly 30 years, and still in many ways do, investigating a couple of basic problems. So it turns out that biological membranes contain lots of lipids that if you just extract them from a membrane and then put them back into water, they adopt non-bilayer structures. And so structures such as this, they have nothing to do with the lipid bilayer. So it's why are they there? Why are these? What functional roles do they have? So that's the question we started off asking in the 70s. Another problem is that biological membranes have asymmetric transviral distributions of lipids. So the lipid composition on one side of the membrane is different than the lipid composition on the other side. And of course, you have ion gradients across the membrane. And so we asked the question, well, can we generate lipid asymmetry in response to ion gradients? And what are the consequences of flipping lipids across a membrane in terms of the structural and morphological features of those membranes? So what we discovered, and this was quite early on, we discovered that we could use phosphorus NMR, where the phospholipid, of course, is a phosphorus in the head group. And we could use that to study lipid polymorphism, or this ability of lipids to adopt different structures. So you just add water, some lipids will adopt a bilayer. And what we showed was that phospholipids will show a phosphorus spectrum with a low field shoulder and high field peak. So it's quite distinctive. On the other hand, if the motional averaging around these cylinders, one gets this peak with reversed asymmetry. So there's a low field peak and a high field shoulder. That's about half as broad. And then if we had isotropic motion, then we get a much narrower line shape. But the bottom line was that we could detect which structures lipids adopted when they're in an aqueous environment. So me and Ben de Kruif, a Dutch scientist, as a postdoc with me in Oxford, proposed a shape hypothesis. So lipids that adopt a bilayer have a more cylindrical shape, and lipids that adopt the hexagonal phase have a more of a cone shape, where the area in the head group region is smaller than the area subtended by the acyl chains. We then, of course, asked, well, what are the functional roles of these lipids? And in order to investigate this, we had to develop very simple systems. You can't. The biological membranes are way too complicated to try and study these properties in situ, and so we had to develop these very simple systems. These are liposomal and nanoparticle systems. And so we developed an apparatus. We invented an apparatus to produce these systems. So we could get we made large structures just by mixing lipids and water and force them through a polycarbonate filter with a 100 nanometer pore size. And out the other end would come these very uniform lipid vesicles, as seen here by cryo-electron microscopy. So we could make these, we could make these things, we can make them of any lipids that we wanted, and then we could start to study the properties. And what we showed was that if we had non-bilayer lipids, or say we had lipids that most of them wanted to adopt a bilayer structure, but if we added some lipids that adopted these other structures, we could generate membrane fusion between these vesicles in a very controlled way. And so, of course, membrane fusion is a pretty important function in biology. Now, we also use these liposomes to look at the consequences of lipid asymmetry. And in order to do this, we synthesized what we termed was an ionizable cationic lipid. This is DODAP. This had the property that at, say, neutral pH, it's positively charged, it's protonated, so this is a tertiary amine. But at higher pHs, say around pH 7, it's net neutral. And so the lipid in the neutral form can flip across the membrane very easily. And so if we set up a pH gradient across a vesicle, they would come to the inside, get protonated and positively charged, and not be able to get back out because charged molecules can't permeate through lipid bilayers very easily. So we could drive lipid asymmetry. We were using this to look at some of the consequences of asymmetric transviral distributions of lipids. But we got a bit distracted. We got distracted by the drug delivery potential. We found we could load cancer drugs into these liposomes exhibiting a pH gradient. Now, a lot of cancer drugs are weak bases. They can exist in a neutral form or a protonated or a charged form. This is looking at doxorubicin, loading doxorubicin. We can load it into such high concentrations that would precipitate on the inside. So these are doxorubicin crystals on the inside of these. This is a 100 nanometer bar of these nanoparticles. So we started a company, and this is 5 of us, 4 postdocs and myself, in 1985 to use these liposomes to improve delivery of cancer drugs to tumors. And actually, this is one of the reasons why we've been successful. We used the company in a very basic way to keep a team together. So we had continuous. We could build on the expertise that we developed. Now, in the middle 90s, we had to start working on gene therapies. This is Jim Miller, who was the CEO of INEX at the time. But one day he came to me and said he couldn't raise money putting old drugs into liposomes, these cancer drugs. We needed to be doing gene therapy, which was becoming quite topical at that time. So this started us off on a whole new path. Now, what is gene therapy? You'll see various definitions of that. Some people consider it's altering the genome, but there is a more general definition, the therapeutic delivery of nucleic acid into a patient's cells as a drug to treat disease. Now, this requires a delivery system. As you know, nucleic acid polymers are rapidly degraded. If you inject them, they don't preferentially accumulate in target tissue. And they can't get into target cells even if they get to the target tissue. So you've got to have some sort of delivery system that avoids these problems. Now, we focused on delivery systems for RNA-based molecules to either inhibit gene expression or promote gene expression. And so this is short interfering RNA to inhibit production of a pathogenic protein. This is basically degrading the mRNA that's produced from the DNA in a very specific way according to the sequence of the siRNA. Similarly, if we can get messenger RNA inside a cell into the cytoplasm, to the ribosome, then it's going to be made into a protein. Now, this is rather remarkable because if we can use RNA molecules, siRNA or mRNA, we can potentially treat most diseases. So it's, and I'll point that out towards the end of the talk. Again, these are large molecules. siRNA is, say, 13 kilodaltons. It's a 21-mer. mRNA, of course, can be, thousands of bases long, and so is a much larger molecule. So we spent the last 25 years developing lipid nanoparticles, or LNPs, to deliver siRNA and mRNA into cells in vivo. So this is still going on, and we're still getting better and better at it. So I'll talk a little bit about some of the early work here. I'll talk about the Onpattro or patisiran story. This is the development of a lipid nanoparticle drug to treat a condition known as hereditary amyloid transthyretin amyloidosis. And then I'll go on to talk a little bit about the vaccine. And I'll close by talking about some of the remarkable additional applications that are coming into focus. So our aim in 1995 was to develop a delivery system to enable a small interfering RNA to silence a gene in the liver. So this was a pretty tall order. We first of all had to package the nucleic acid in the lipid nanoparticle, inject it intravenously, have it go to the liver, and then get out of the circulation, be taken up by hepatocytes, and then get out of the endosome and deliver the siRNA to the interior of the cell. This is a pretty big challenge. Nature goes to great lengths. Your immune system is exquisitely designed to make sure we don't get foreign genetic material coming into our cells. Our first problem that we hit right off the bat was that if you want to encapsulate these very negatively charged nucleic acid polymers, we had to use cationic or positively charged lipids to form some sort of a hydrophobic complex, as is indicated here. Now, there's no positively charged lipids in nature. They're really toxic molecules. There's only net neutral lipids or negatively charged lipids. And so what we did was we said, okay, we synthesized this ionizable cationic lipid for our lipid asymmetry work. And it had this property that at low pH, it was protonated and positively charged, but at neutral pH, of course, it was relatively uncharged. And so we wondered whether we could encapsulate, let's say, the pH 4, which is the acidity of a lemon, so it's not really that acid. Could we encapsulate in a medium at pH 4, and would the contents be retained when we move the pH up to physiological pH? And what we found was that we could load nucleic acids, DNA, RNA, into lipid nanoparticles by, first of all, having the nucleic acid in water at pH 4, And then all the lipids, and this is, say, the green one is an ionizable, the ionizable cationic lipid, and we have a couple of other structural lipids like cholesterol and phosphatidylcholine in there as well, along with the PEG lipid. This is an ethanol. We mix these together very rapidly. Well, one of the first things to fall out of solution is the oligonucleotide surrounded by the positively charged lipid. And so you can see intuitively how it is that you can get fairly efficient encapsulation. If you do this rapidly enough, then the PEG lipids fall out and coat the outside, and you can get these very small systems resulting from it. And then you dialyze away the residual ethanol and raise the pH. And so this turned out to be a really good way of developing a delivery system for siRNA. Importantly, is relatively non-toxic. And so we can now have systems where at least there was a possibility of using them in an in vivo setting. You have a hydrophobic core, and then they're well suited to encapsulation of negatively charged macromolecules, really quite a great variety of them, but certainly nucleic acids. You get encapsulation efficiencies of 100%, stable and monodispersed. We can vary the size just simply by varying the amount of PEG lipid we put in there, scalable and reproducible. So this is now we had a system that we could actually start to maybe to see whether or not they were reasonable delivery systems for siRNA. Now, so I'll just move on to that program now. So this is small interfering RNA over here, duplex. And I gave a talk at a conference in London in 2004, and I was pursued by Victor Kataliansky, who was a vice president research for a company in Boston called Alnylam Pharmaceuticals. So Alnylam was founded in 2002, and they were founded to use small interfering RNA as a therapeutic to silence or inhibit the production of pathological proteins. And so Victor said, well, we have a delivery problem. How do we get our small interfering RNA into the liver, into hepatocytes in vivo? And so this resulted with a collaboration between my lab as an Acuitas Therapeutics company I co-founded, as well as my UBC lab. This went on for about 7 years between 2005 to 2012. And we synthesized many different lipids. We tested many different nanoparticle formulations, and it was a very, very successful program. Resulted in a lipid nanoparticle system that with silenced genes in the liver, with the lipid nanoparticle containing siRNA with a very high therapeutic index. So what we did was we started with the question, do these systems, can these lipid nanoparticle RNA systems containing these ionizable lipids silence genes in the liver following an IV injection? Now, the lipid nanoparticles get into cells by endocytosis. This is probably a process which many of you are familiar with. And so they're taken up. They progress to if they don't get out of the endosome, they're going to be processed by lysosomes, and so therefore, of course, it's going to degrade the mRNA. So what we have to do is to get out of the endosome at some vulnerable point to allow the siRNA to escape. So we need to destabilize the endosome and release the nucleic acid into the cytoplasm. So how can ionizable lipids destabilize endosomes? And this is another example where basic research turned out to be quite useful. What we found was that positively charged lipids combined with negatively charged lipids found in biological membranes to induce membrane disruptive non-bilayer structures. And so if we have an anionic lipid that's present in a biological membrane, such as phosphatidylserine, negatively charged lipid, if we add a cationic lipid, it flips it straight over to these non-bilayer structures, and so therefore it's very membrane disruptive. This week, we can attribute this to the kind of a cone shape that results from the electrostatic attraction between the head groups. So really, we came back to lipid polymorphism, these very basic structures that we've been doing many years before. And so we designed the ionizable lipids so that they became protonated or positively, they're net neutral at neutral pH, but as the acidity of the endosome increases, there's inward directed proton pumps, then they become positively charged. And we designed them to have maximum ability to disrupt the membrane and release the oligonucleotide on the outside. So we had a rationale that we could use to improve the design of these systems. Now, we assessed the in vivo potency using a factor 7 model. Factor 7 is a clotting protein made in the liver, and so we could make siRNA against factor 7, inject it intravenously, and then assay for factor 7 in the blood 24 hours later. And if they were very good at knocking down factor 7 in hepatocytes, then of course they wouldn't detect much factor 7. So it was convenient. We could do two experiments a week when we were trying to optimize these systems. We found that the potency was extremely sensitive to the species of the cationic lipid that was employed. We synthesized, this was a great chemistry program. We synthesized over 300 ionizable lipids and moved through sequences such as this. Now, these lipids don't look all that different. So they all have tertiary amines, but they have very, very different activities. And what we found was that the potency was highly sensitive to the pKa, the point at which these positively charged lipids got protonated, as you can see here. And this is a log plot. So the potency, which is 1 over the ED50, the ED50 is the dose required to induce 50% gene silencing. But you can see 3 orders of magnitude if we move as little as half a pH unit away from this optimum. And so this was we still don't know exactly what's going on here, but it certainly is a phenomenon that we could use. So this is just looking at factor 7 expression. And what we did was we moved from a system where we could see silencing at 10 mg per kg of siRNA down to 5 micrograms of siRNA per kilogram body weight. And this was without increasing the toxicity. So we had a huge therapeutic window of 1,000. And so this was the point at which we decided, okay, these things are good enough to go into the clinic. So one of the things about a gene therapy or genetic drug approach is the generality. We can silence factor 7, but that means we can silence any gene we wanted to. And so the clinicians at Alnylam were so, okay, well, what gene should be silenced and what disease should we go after? And so they chose to develop a lipid nanoparticle therapeutic, siRNA therapeutic, to treat a disease known as transthyretin-induced amyloidosis. There's about 50,000 people worldwide that suffer from this. So transthyretin, it's a protein made in the liver, and it's a retinol binding protein. And if there's mutations in it, it's a large protein, then it often tends to form fibrils in the blood, and these amyloid fibrils will deposit all over the body, and they have really nasty effects in nerve tissue and in the heart. And so it's a neurotoxic. There's neuropathies that result, and also cardiac problems, usually fatal within 5 years of diagnosis. And as you can see here, this is really a nasty disease. It's a wasting disease. And of course, very nasty if you start to stumble in your early 30s, you kind of know what you're in for because a relative will have gone through this process and you'll be familiar with it. Anyway, so a very simple therapeutic approach here. That's just stop transthyretin being made in the liver. And so making siRNA, put in that nanoparticle, and then the idea is we have less tetramers, the deposition is prevented, perhaps we could get clearance and improvement, and so it's potentially a very simple solution to a devastating disease. this is really part of the attraction of these gene therapies, is the ease with which you can design your medicine. There was a phase 3 clinical trial, 148 people that were administered the drug, which at that point was called patisiran, or the placebo group, 77 got sterile saline. And the endpoints of this trial were neural impairment score, disability, ability to walk, quality of life self-reported, body mass, and so on. Now, what was shown over the 18-month course of this trial was that the group on the placebo got steadily worse according to the neural impairment score, whereas remarkably, people suffering from this genetic disease started to get better. So this is quite startling. There's an improvement in symptoms for people suffering from this devastating hereditary disease. And the phase 3 results were announced in 2017. So it hit the primary endpoint and all secondary endpoints. So really with remarkable p-values, 10 to the minus 24, I will say that's 1 over Avogadro's number, but there's absolutely no doubt that this drug works. And all the secondary endpoints, again, very convincing p-values. So it's really a relatively curative therapy for what was previously a fatal disease. There's patients been on this drug now for 8 or 9 years, and they're continuing to benefit. So this was approved by the FDA in 2018 for treatment of transthyretin-induced amyloidosis. It was the first FDA approval of an siRNA-based gene therapy drug, and really a clinical validation of the lipid nanoparticle delivery system. So it's a pretty big deal. Really demonstrates the power of gene therapies. So now I'm going to go on to the story of the vaccine. And I'll point out here the serendipity of this process. So we asked the question, and this was with a company that I co-founded called Acuitas: does this lipid nanoparticle technology that we developed for small interfering RNA enable mRNA-based gene therapy? So in other words, we had a system where we were silencing a gene in hepatocytes. Well, if we put mRNA in there, would it express a gene in hepatocytes? And so this was, this was a bit of a long shot when you think about it, because the mRNA is, of course, so much bigger than the siRNA. What we found was that we could design these lipid nanoparticle systems and use the liver as a bioreactor to produce any protein you want. Now, think about that. It's really quite remarkable. I mean, your liver produces 70% of the protein in your body, and so it's a really good producer of protein. In this case, what we're doing is we're administering mRNA coding for erythropoietin, and so these get supramolecular levels of erythropoietin in the circulation about 6 hours later. This is in a pig model, so a large animal. This was from the work we were doing. This was obviously, there's so many diseases that are now approachable. That we could produce a protein that, whether everything from Tay-Sachs to various metabolic disorders anyway, there's a huge number. You can make a monoclonal antibody against a cancer. There's all kinds of possibilities where you can use the liver as a bioreactor to produce the proteins you want. But this is where serendipity comes in. We were approached by Drew Weissman from the University of Pennsylvania and Catalina Carrico of BioNTech, and they needed a delivery system for their mRNA vaccines. They worked for many years together to explore the potential of mRNA as a vaccine. They came up with a modified mRNA. In vitro, they could reduce immune activation and increase gene expression, but they needed a delivery system in vivo. So we have a delivery problem. This is like Victor Kataliansky and Alnylam in 2004. So we have a delivery problem. How do we get mRNA coding for viral proteins into muscle and immune cells in vivo? And so this is an IM injection, so we want to transfect muscle cells and antigen-presenting cells to get an MHC1, MHC2 class response. Now we weren't immunologists, but Drew is, and so the study was first of all done on to develop a Zika virus vaccine. So here, instead of having mRNA coding for some therapeutic protein, we had mRNA coding for the Zika virus pre-membrane and envelope glycoprotein. And so this can be made in a matter of a couple of months, and then we could encapsulate it in a couple of days or a day or so, and then test this in animal models. And so this is an IV intradermal administration, injecting the lipid nanoparticle containing mRNA coding for this Zika virus protein at time 0, and then challenging with 2 weeks or 20 weeks with a lethal dose of the Zika virus. And so as you can see here, complete protection for the immunized animals against infection of both 2 weeks and 20 weeks. So really quite a remarkable result that really proved that these systems made excellent vaccines. Now, this triggered interest from a variety of sources, and one of them was BioNTech, and partnered with Acuitas to actually looking at to develop influenza vaccines. And BioNTech was also working with Pfizer on a flu vaccine. And of course, in January 2020, all efforts switched to a COVID, to making a COVID-19 instead of a flu vaccine in January, February of 2020. Now the rest of this is history. So this trial went very, very quickly. It was announced in November of 2020 that this vaccine was 95% effective to prevent COVID-19, consistent across age and gender and ethnicity, relatively well tolerated. And at that point, they were projecting, saying they were going to make 1.3 billion. I think they were closer to 3 billion doses by the end of 2021. And as we know, this is playing a major role in controlling the COVID-19 pandemic. But so as I say, pointing out, we were trying to produce proteins in the liver, and as a result of meeting with Drew Weissman, suddenly found that we developed rather effective vaccines. So it's been a really quite an amazing journey. From these very basic studies of, as I said, lipid asymmetry and lipid polymorphism, to developing medicines that have really really amazing applications as vaccines and other possibilities. And so really this journey is just beginning. This is, I think that these lipid nanoparticle mRNA systems are really going to constitute the third generation of pharmaceuticals. First generation of drugs, of course, is small molecule drugs. Second generation are biologics. But this third generation, really mRNA gene therapies, plus protein replacement, vaccines, and gene editing. So we really have all of those possibilities. We can have protein expression. We have the vaccine application. We can use CRISPR and other editing techniques to basically knock proteins out. So let's give a few examples of this that have appeared in the literature in the last while. This is what appears to be a universal flu vaccine. So here's a lipid nanoparticle containing. You can see these. This is looking at protection by looking at body weight following an injection of these viruses, of these flu viruses, from a variety of different sources. One from 1934, 1999, etc. So these are very different viruses. So they contain a variety of mRNAs coding for hemagglutinin, coding for neuraminidase on the influenza virus, a channel protein, a nuclear protein. You can see total protection. So it may well be that quite soon we're going to see a universal flu vaccine, so we don't have to keep on taking it every year, which would be rather nice. Treatment for cardiovascular disease. This is a lipid nanoparticle. In this case, of course, the previous one was administered intramuscularly. This is a lipid nanoparticle containing a CRISPR adenine base editor and a PCSK9 guide RNA. So they're basically not gene editing PCSK9. This is involved in regulation of LDL levels. And as you can see here, this is in non-human primates, but the results are carrying through into the clinic, basically knocking down the production or the amount of low-density cholesterol in the circulation at will, with very extended lifetimes. This is an IV injection. We're doing gene editing in this case in hepatocytes. So it's essentially a vaccination against atherosclerosis. Well, so atherosclerosis is obviously a huge killer, rather remarkable result. But a treatment for heart failure. And so in this case, it's a lipid nanoparticle containing mRNA coding for CAR, for chimeric antigen receptor against fibroblast activation protein, which is associated with the cardiac injury, and then targeted T cells with antibodies against CD5, which is a marker for T cells. So again, you can see that if we induce a cardiac injury, this is the control, induced cardiac injury, you can see the fibrosis resulting as a result of this therapy. You can reduce that fibrosis significantly. The ejection fraction comes back to pretty much normal. Really remarkable results that go on and on. Treatment for obesity or metabolic disease. Here we have a lipid nanoparticle containing mRNA that's encoding a growth factor FGF21 administered IV and expressed in the liver. And in this case, we're reducing the body weight if they're fed a diet-induced a high food intake or high fat intake. Also reduces plasma triglycerides and cholesterol. So these are again treatments for cancer. So in this case, an mRNA coding for a bispecific antibody that basically pulls a T cell into close proximity with a tumor cell, promotes production of or promotes elimination of large tumors in mice. I'm just going through these just to give, as I say, a little bit of the flavor. For the transthyretin-induced amyloidosis, of course, this is done by a company called Intellia, based in Boston. So in this case, again, a CRISPR-based approach to knock out transthyretin. And as you can see, they can get from one injection, get relatively complete knockdown of transthyretin. So this is going to provide some rather significant competition for the Onpattro product. A rare disease, the treatment for I'm just giving these as examples, glycogen storage disease. So LNP containing mRNA coding for glucose 6-phosphatase for protein replacements. And here again, getting treatment, let's say 1 mg per kg, an IV dose. You're seeing basically that they're really getting back to normal in terms of their blood sugar levels and, of course, preventing liver damage. A treatment for a condition known as propionic acidemia. This is LNP with mRNA coding for propionyl CoA decarboxylase. And so this is an effective treatment for this rare disease. The list goes on. And so one of the things, as I mentioned at the outset, that's really remarkable here. It's not in the small molecule, like it is in the small molecule arena, where we have to spend $1 billion in 15 years to develop a drug. Once we identify the protein we want to silence or express or edit, we can produce the SR and mRNA in a matter of a few weeks. We can encapsulate in the lipid nanoparticle in a matter of one day or less. And so we have these highly targeted personalized medicines available in real time, in time on the order of weeks. So it's really going to enable a multitude of gene therapies. So I think, as I mentioned, with the next generation of pharmaceuticals, ranging for obviously as huge applications now as vaccines, chronic diseases, as I tried to indicate. These systems transfect neurons, microglial cells, et cetera, very well. Inherited diseases, the list goes on and on. Now, one of the points I just want to close on making is that this is obviously the work of probably close to 1,000 people over the years. But I've had very close associations with Mick Hope and Tom Madden. I've worked with them for over 40 years, and it's been one of the things that's made us very successful is this long-term association. Steve Ansel, Ying Tam, Barb Mui, Paula Lin, Sean Semple, more than 20 years. Similarly for many others here. The Alnylam group, Mark Tracy, Akina Kink, this was a pivotal collaboration. Marco Cipollini, professor in the chemistry department at UBC, played a pivotal role in the design of these ionizable lipids. My own group, and of course, Drew Weissman at the University of Pennsylvania. So with that, I'll close and take any questions that anybody may have.
Thank you, Dr. Cullis, for the wonderful speech. Now it's time to move on to the panel discussion and Q&A sections. Please remember to scan the QR code and raise your questions on the slide link. Ladies and gentlemen, I would like to invite to the stage the moderator of this forum, Dr. Gao, and panelist Dr. Wu Hanzhong, Director of Biomedical Translation Research Center of Academia Sinica, Dr. Qiu Zhenxun, Vice Superintendent and Professor of Division of Pediatric Infectious Disease of Linkou Changge Memorial Hospital, Dr. Ye Changjing, Chairman and CEO of Onward Therapeutics SA, and also the 2022 Tom Prize laureate, Dr. Peter Cullis, to join us online. Welcome. Next, we'll leave the time to the moderator.
Thank you so much. First of all, thank you so much, Peter, for that lovely and very insightful journey through history. I think you have such an incredible story to tell. Let me kind of set the stage for what's happening here. For the next 30 minutes, we will have a discussion amongst the panelists and Dr. Cullis. And then we will then break into a Q&A session. I just want everybody to know that Dr. Ye would need to leave by 12 noon, so we'll make sure that we stay on time for that. And then, as the MC already mentioned, the Q&A session will involve questions. We'll be taking questions from the audience. So that's the second half of this session. And we're delighted to have on the stage Dr. Wu, who will really represent a perspective from translational research. And particularly, I'm interested to hear on the role of policymakers on the translational journey of biomedical technology. We also have Dr. Ye. She is a renowned scientist of her own right, but also an industrialist. So she will provide insights on how industries can bridge fundamental research and clinical applications and the many challenges that industry faces, including fundraising, regulatory approval, and those kind of very critical stages. And we have Dr. Chu, who will obviously provide critical insight on the end users of these technologies. So from clinical perspective and from patient's perspective, how new technology can address these clinical pain points. I have so many questions myself, and as the moderator and chair, I will take the prerogative of asking Peter the first question, which is, as you were talking about the history of your own journey, it took me back to when I was a graduate student 20, 30 some years ago. And I remember also when I joined the faculty in the School of Pharmacy in Wisconsin, there are a few colleagues, including Tim Heath, maybe you know, he works in lipids, and was also in Oxford, I think around the time when you were there. So my question is really about, we have seen a lot about material platform technology come and went, really in the drug delivery space, and lipids have been around for quite a long time. So what gives you the staying power to sustain this kind of level of research excellence when there are these fashionable platforms that came and went, and obviously your staying power proved them all wrong, that you were super successful in developing what the world needs. So can you just comment on that courage, that stamina, that desire to continue?
Yeah, well, there's a couple of components to that. Obviously, one of them is the fascination with the basic problems of, you know, as I said, lipid polymorphism, lipid asymmetry, the roles of lipids in membranes. And so in many ways, that's what still, you know, drives me. It's quite incredible, you know, it's quite incredible. Even now, after 50 years of studying lipids and liposomes and all these systems, we still don't really understand you know, all the ways in which this approach can be used. Nature doesn't choose molecules by accident. That's one thing you learn. You know, these lipids have remarkable properties. I'll give you a little bit of an example. I mean, okay, we found ways of encapsulating, say, small molecule drugs, and we found ways of encapsulating you know, the nucleic acid-based drugs, and then we found ways of using those as delivery systems for therapeutics. But the flexibility, I mean, what nature does with membranes is, of course, we have cell membranes, and inside that we have organelles, and we have all kinds of things going on inside a cell. And so you start to wonder, well, can we make these lipid nanoparticles more complicated? Well, you can. What we're finding now is we can say, if we add triolein, we can have something that has a fatty component and then an aqueous component, and we can put different things in there and start to give them make hybrids that have a variety of functions. I mean, for example, we can start putting in gold nanoparticles that might be sensitive to laser light, along with small molecules, and then attempt to blow them up in particular regions. But these are the kinds of things that keep me fascinated. Simply because you know, we're only just touching the surface of the properties of these molecules. They're absolutely marvelous, and I've never tired of thinking about them. That's for sure.
Great, thank you, Peter. So I guess instead of chasing after fashion, you chase after curiosity. Is that sense of wonder and what's next that kept you going, and that's actually, I think, a good lessons for all of us to remember. So I will have Dr. Wu ask the next question.
Okay, yeah, thank you, Tom Price Foundation and President Gao, kind introduction. So yeah, also thank you very much for Professor Cooley's give such a wonderful and informative talk. Okay, so your speech is very, very inspiring to us. I'm Han Zhongwu, the Director of Translational Research Center, BioTrack, in the National Biotechnology Research Park, NBRP. Okay, so I would like to provide a few comments and my opinion on Professor Cooley's outstanding research. So the lipid nanoparticle, LNP, are essential for the delivery of mRNA vaccine and the drugs. Professor Cooley is the best known for the development of ionizable lipid, LNP. This type of lipid markedly reduce the toxicity and increase the biological function of the cargo, like mRNA. Furthermore, Professor Cooley has also able to overcome a very difficult problem we so-called is endosome escape of the cargo, like the mRNA. These breakthroughs are essential and critical to the function of the COVID-19 mRNA vaccine, and are rapidly enabling the clinical achievement of gene therapy, as well as other life-saving medicine and the drugs. It has also opened up a new era for nucleic acid technology in the prevention and the treatment of many diseases. So as mentioned by the Professor Cooley, yeah, currently clinical applications are focused on using the mRNA LNP for vaccination, protein replacement therapy, and treatment of genetic disease and so on. Okay, so Professor Cooley's LNP technology has translated in the field from the academia to biotech, as his extensive basic research knowledge, including the physics, chemistry, and the biology. His story clearly demonstrated how multidisciplinary basic research provides a foundation for the applied science and the biotechnology. So, Professor Cooley, your tremendous contribution in this field deserve our utmost respect and appreciation.
Well, thank you.
I have a couple of questions, and maybe have a further time I will ask the other question, but I only one question. Just want to ask. My question is about the cytotoxicity of the lipid nanoparticle. Okay, as we know, several pegylated drugs has been shown cause allergic effect, and also secretion of inflammatory cytokine has been associated with use of the lipid nanoparticle in mRNA vaccine. Could you please comment on this issue, or suggest how to resolve this concern? So in the future, which direction do you think will be particularly important in development new ionizable lipids? So the other, what are other biomaterial besides LNP do you think might have a clinical potential for delivery of the drug, nucleic drug? Okay, yeah, that's all my comment and my question. Thank you.
Okay, thank you. Just with regard to the PEG lipid, the polyethylene glycol lipid, the reaction, I mean, there obviously have been reactions which have been associated with the presence of the PEG lipid. They don't seem to be all that prevalent. You know, it's a little, we need the PEG lipid there to stabilize the size of these particles in storage. Actually, we've designed the PEG lipid so that it comes off. It actually, when it's in an in vivo situation, it diffuses from the membrane quite rapidly, by the time on the order of a couple of minutes. and so it's no longer associated with the particle. We have to do that, otherwise these systems aren't transfection competent. They won't be taken up by cells if they have a PEG coating. Anyway, so how we might counter allergic reactions to these lipids, I think the jury is still out on that as exactly how we're going to do it. Because you run into this problem that any other compound that we put on the outside to control the size may be worse. And now we have a medicine vaccine that's been in literally billions of arms. We know how to counteract any of the effects that may occur. Anyway, it's going to be difficult to get a new coating substance that's going to be as accepted perhaps as the PEG coating. So we've been thinking about it. One of the ways that we're going at it is to see whether or not we can just eliminate any PEG coating at all, any of these the polymer-based systems. And it looks like we might be able to by increasing the amount of the phosphatidylcholine that's present. But this is very early days, and the stability of these systems is not great. And so but of course with vaccines, it's basically stability with regard to size. So with vaccines, you're not quite so worried about that as you are, say, for an IV injection. Now, the other question you were asking was about the cytotoxicity of the ionizable lipid, and the variable, the most important variable actually is the pKa of the ionizable lipid. The closer, if that's much above 6.4, the closer it gets to neutrality, the more toxic these molecules become. So, in other words, they're partially positively charged at neutral pH, and that's where you start to see toxic side effects. But if you hold to that range, they seem to be pretty well tolerated. And as I mentioned, the patients that are taking Onpattro, this is the drug to silence transthyretin in the liver, have been taking really much higher dose than for the vaccines every 3 weeks for now eight years or so, and really are not showing any signs of there hasn't been any toxicity signs that have been associated with the ionizable lipids. So I'm optimistic that's an ionizable lipid that's not very easily broken down in the body. We're now getting much better at developing ionizable lipids that are broken down more quickly, and so therefore don't hang around in the body for too long. But I'm optimistic that the toxicity of the ionizable lipid is something that we can control.
Right. I think it raises a very interesting perspective, is that life is a balance. You're balancing toxicity, bioavailability, stability, and obviously translational of technology. You think about scalability, cost, and regulatory. So there's no, when you put everything together, how to hit that sweet spot, and what's the most efficacious way to benefit most people, it's not a simple answer. So Peter, I think we all know the challenge there. Grace?
Hello, good morning. Oh, actually, good evening for you, Professor Collis, and good morning to the audience here in the auditorium and online. I'm really impressed with what you have done with 50 years of lipids. I'm Grace Ye. I'm trained as an immunologist, but then I did my postdoc work in Europe and went to join biotech in Boston for 15 years. And then I returned to Taiwan. I founded a company called Pharma Engine. And it just so happened that my project, liposome irinotecan, is really, really very close to you. And this was 2003. And I was the founder, CEO, and president. Fortunately, you know, I didn't say to my people, do not develop liposome formulation for old drugs, because I think that was happened to you at INEX. So we carry on, and eventually we developed a, I think it's different from your formulation. It is a negatively charged, and through the use of TEA, SOS, triacyl ammonium sucrose sulfate, octosulfate. So this we create a gradient to move irinotecan, which is a very hypophilic drug, into the liposome and protected it against the inactivation of various enzymes. And then it also has the so-called EPR effect when to accumulate in the tumor. And in this way, that the drug released on site and then catalyzed by the drug to become an active metabolite, SN38. So it's a very well thought of drug, but eventually it became a medicine for the treatment of pancreatic cancer, metastatic pancreatic cancer, and it's approved in 2015. So when I hear what you said about the lipids, we certainly have a battle with the lipids during the laboratory and also scale up. I think that the scale up is not an easy task. And so my question is that you have demonstrated the lipid formulation can be applicable to so many diseases. And do you see that there's some very challenging therapeutic areas that for the lipid formulation, you will have certain limits? And so this, because you showed us many genetic diseases, even cancer, cardiovascular diseases. So is there anything that you think that your formulation or your technology will face some challenges? And if so, for gene therapy, people also talk about should we have a on-off switch? I think this is also balance the benefit toxicity ratio. Thank you.
Yeah. Well, that was a very interesting story you told about irinotecan. It's a great drug. It's one that we've played around with that a little bit too. But it is ideally suited in many ways to a lipid-based delivery system. Just with regard to problems, associated with lipid nanoparticles. They're big structures, at least on in terms of things that might get across various barriers in the body, in particular the blood-brain barrier. And so the ability to get into the brain, I think from an IV injection, it's going to be a long time before we solve that one. I suppose there's things like ultrasound that could get local leakage. So I think the first applications are going to come in by direct injection, whether intrathecal or intraventricularly, and I think that's going to be quite, as I said, these systems transfect neurons quite well. The reason for that is that the way that these nanoparticles are taken up by the liver is that they associate with apolipoprotein E, and that triggers uptake into hepatocytes. Well, ApoE is the major lipoprotein in the brain, and so we're dealing with a very similar mechanism there. So they're highly effective in brain tissue. So I think that's going to be the start. Similarly with the eye, you're going to see direct injection. There's already many reports that are coming out in that arena. With regard to the on-off switch, I totally agree with you, and it's one of the things there's actually a benefit in some ways with the mRNA approach because, mRNA is only expressed in a transitory way. And so you do have an off switch automatically, which in some cases, you can go the gene editing route and knock something out relatively permanently, but at least you can titrate it and say knock it down 50% if it's LDL or whatever. This is the RNA-based drugs are pretty safe in that sense. I mean, they don't get into the nucleus, and they don't unless you have a CRISPR approach, a gene editing approach. They don't really affect the genomic material. So it's when we presumably one day we'll be able to do. The gene therapy on the genome itself in a safe manner, but I think we're a long way away from that. And in the interim, the RNA approach allows us to do almost everything you could do. It's just you have to keep on injecting, and I think that's both that could be viewed as being a safety thing, a safety benefit that's really quite important.
Just a small comment. Indeed, the blood-brain barrier it's really a big challenge because we use Onivyde, this formulation of irinotecan, and we tried in intracranial injection into dogs, and it worked. This is a brain tumor model, but then we tried IV, it's not so well. So certainly it has some limitations. Thank you.
Yeah. Thank you.
I guess it's safe to say that biomedical technology and health industry is an industry that will never sunset because there are just so many diseases with so many brilliant solutions that require so many innovative ideas to come and work together. I think that was another key messages Peter gave everybody here, is that teamwork is absolutely important. That no one person can do everything. That if we want to make the world a better place, at least, we have to learn how to work together. So thank you, Peter, for that very critical message, teamwork. Dr. Chu, yes?
First, I would like to thank Town Prize Committee for the invitation. So I have this honor to talk to Professor Coolidge online. I actually contacted you before. I was a postdoc in UBC from 97 to 99, studying liposome-embedded glycans to treat complications of cystic fibrosis. So my question is very simple about cystic fibrosis. Actually, I'm a physician with specialties in pediatrics and infectious diseases. So as you said in your presentation, almost all diseases can be treated by gene therapy or LNP-based mRNA delivery system. So actually, apart from the rare diseases you have mentioned, the disease always in my mind is cystic fibrosis. The CFTR gene actually was first discovered by Professor Choi, who later in a hospital for sick children in University of Toronto in 1987. And Professor Choi actually became the president of Hong Kong U later. And after so many years, the discovery of the defect gene, there's still no specific therapy available to halt the progression of the diseases. So my question is very simple. Actually, you mentioned the disease in your presentation. Do you think the technology you developed will sufficiently to improve the treatment outcome of cystic fibrosis gene therapy?
That's a tough one. There's, obviously there's a lot of interest in that. But you do have this problem of if you go in, an aerosol, there's a mucus layer that in terms of penetration poses a problem. If you go in IV, we're still not very good at transfecting the lungs without deleterious effects. So and then added to that, CFTR gene is huge. You know, which compounds the issue. So yeah, it's gonna be I mean, I know that people are working on this. We have done some work with aerosols. The variability there is a real problem. You have to, you know, anyway, there's to chew your way through the mucus layer. I have a feeling IV is probably in the end going to be the technique that gets us there. But one of the issues that we're going to find, I mean, we're working now with systems that circulate for much longer than, say, the systems which go to the liver. The Onpattro or the type formulations are cleared from the circulation within 30 minutes, and 90% goes to the liver. So there really isn't much chance for other tissues to be transfected. And so what we're doing is developing systems that are transfection competent, but that will circulate for a time on the order of hours. And there we are starting to see some signals, but they're not very strong from other tissues. Well, we get pretty good at bone marrow, but the lung and kidney and others, we can see some signals, but they're not huge yet. And it's going to take a while before we get, but in the end, probably what we'll be aiming for is a more global gene therapy. Then we'll have to try and figure out how to turn things on and off in particular locations. That can be done theoretically by some molecular biology approach, perhaps, or some, carrying along some agent along with the lipid nanoparticle that we can turn on and off using, say, external radiation, X-rays, or laser, or RF. So all of those, the flexibility of the lipid nanoparticle is that we can incorporate those kind of things into the particle in a reasonably scalable manner. But it's early days. The first stage is going to be to actually see, well, can we transfect, even though we're transfecting other tissues, can we transfect lung? Second, can we express CFTR? Does it have deleterious effects in the rest of the body, which it might? Anyway, so it's a great target, though. I mean, that's for sure. And it is one that we're trying to get a collaboration going, but there's in that area. So it may be something we should talk to you about. Because the potential is, obviously, I worked in a CFTR lab in London for 6 months, and so I know the problem.
Thank you, Peter. I think as you were answering the question, it kind of struck me that when all of us are faced with any kind of challenges, life challenge, research challenge, it's so easy to just think about what cannot be done. That would be a very typical first response, like, oh, that's too hard, or I don't have this equipment, or I cannot do that, I don't know this. But you were so positive in answering that question. You definitely gave us something to think about, which is what is possible? What could I try? I think just like your very first few slides, that if you don't have it, build it. If you need it, get on with it. Build something you don't have. I think, we are so caught up on, well, I don't have this resource. I don't know this. I cannot do that. But if you have that positive energy, that positive drive, many things, I think all things become possible. I think for all the young researchers and not so young researchers like ourselves, these are messages, these are reminders that we have to take with us for a very long time. So thank you, Peter, for that dose of positivity. Very Canadian. Thank you. I think in the interest of time, Grace has 5 more minutes, so I turn the mic over to Grace.
Sure.
Yeah.
Thank you. I would like to ask you, because you have developed so many formulations of various type of projects, when you did your screening for your maybe hundreds of formulation, what do you think are the key parameters, and how do you, what's your I think the logic of a flow chart to you that eventually you have a concept for the formulation all the way to maybe human. So this it involves many different assays, testings, and of course there are also something that's not totally related to experiments. That's the so-called the regulatory process. But in your mind, with your such a vast experience, each time when you take on a project, you always have an end, right? That's the what will be your development candidates. How you think about this formulation will have certain properties that will allow you to move on to the next. Thank you.
Yeah, that's a good question. One thing that is I find crucial is to be able. Biology is very complicated, and if your system, if your delivery system, is not well characterized, and you aren't making exactly the same thing each time, then the results can get incredibly confusing. And so that's certainly the very first thing to be concentrating on, to make sure that your basic system, the size, the amount of drug in it, what time, how long it takes to come out, all those really basic things. Also, you need to be highly efficient, 100% loading, all of those parameters. And before you ever go near an animal, if you start injecting material into animals, and I've seen people doing this that is not well characterized, and then the next formulation, the next batch has some differences, and they'll see some different results, and as a result, things don't move forward. So that's a really big. The other big one is toxicity. Often you're looking at potency, say an anti-cancer potency or whatever disease that you're trying to treat, and one tends to minimize the your therapeutic index, the window that you have. Now, a problem with looking at animal models and then translating that to the clinic is that you very often will lose quite a lot of your therapeutic window as you move to the clinic. You might have a therapeutic window of 10 times. In other words, you give 10 times the drug before you see toxic effects in a mouse model. But you're very likely to find when you get to the clinic that that's only a factor of 2. And so you really need to get that window as large as you can before you get to the to the non-human primates into the clinic, because toxicity will kill a drug every time, and that's a lesson that I think we all learn the hard way. If you take a drug into the clinic, it's and then you start to see toxic side effects. Boy, oh boy, that'll cause problems.
May I have a follow-up question, a short question? I think before you get to toxicity, you always want to establish some kind of relationship between in vitro plasma release versus eventually you go into animals for in vivo pharmacokinetics. I wonder whether you have seen some trend or that's different depending on the various projects.
Well, there's an example. For the Onpattro type product, and it really extends through the vaccine product too. We kind of designed those systems to be a little unstable. And so they're very rapidly accumulating into the liver, and the PEG coating diffuses off, and they're then able to associate with the cell and be taken up into the cell. The design for systems where we, as I think I mentioned, that with longer circulation lifetimes, you have to get systems that are more stable. Now, one of the things that has been relatively ignored in this field with regard to mRNA, it's not so much a real problem for siRNA, but it's the stability of the mRNA in the lipid nanoparticle itself, and how it is you can adjust lipid nanoparticles so it protects the mRNA more adequately in vivo. And so you can get some interesting results there. Obviously, the longer the mRNA stays, there's interesting problems that evolve. For the siRNA systems, the Onpattro type formulations, there's roughly 1,000 copies of siRNA per lipid nanoparticle. And whereas for say the mRNA, you're often down to 2 or 3 copies. I mean, just simply because there isn't, that's the maximum you can pack in and maintain the size. Anyway, so this means that if one of those things gets degraded, you lost 30% of your activity. So whereas, let's say a few of the siRNA, well, they're pretty robust anyway because they're made to be more stable. The chemistry is more stable. Anyway, so protecting the mRNA in these formulations is really quite important, and characterizing that pretty rigorously, as you say, in an in vitro situation in the presence of serum, et cetera, before you go in vivo can lead to some interesting conclusions. Sometimes you see no activity, you might be blaming your ionizable lipid, because it could easily be the stability of the lipid nanoparticle you have is relatively unstable. Anyway, so these are the kinds of variables that we worry about all the time.
Okay, thank you, Peter.
Thank you very much. I have to apologize. I have to leave to go back to Taipei for another meeting. But really, really nice. And I want to thank Tom Price for inviting me and President Gao for chairing this session. Thank you.
Thank you, Grace. Thank you. Okay, so we're moving to the second half of this session, which is taking questions from the audience. If we could have the QR code again, people can enter the question. I have all the questions here on my notepad. And the questions can be for Peter, our speaker, or any of us on the stage. And I was listening to Peter's response to Grace's last question, and Peter mentioned the word, learn things the hard way. And I thought that is so true, because I immediately reminded myself of my own journey, having my own startup companies. And one of the challenges I did not realize was how to remove endotoxin from the final product. So if something works in a laboratory, it doesn't mean it will work the same process as you try to scale this up. And I definitely learned that lesson the hard way. But I'm glad I learned the lesson, because not knowing what you don't know is actually a lot worse than learning lessons the hard way. So I've been looking through all the questions submitted thus far, and I think it would be great if we could have the QR code so they can enter more questions. Speaking of lessons learned, I think this question is for everybody on the stage, Dr. Chu, Dr. Wu, and Peter. And we start with Peter. And the question is, what is the biggest obstacle in your journey so far? It's a big question. It's a big question. And I think the more interesting question could be follows, which is, how did you overcome obstacles and continue to work? I think all of us face that question every day. I'm very curious to hear from Peter, from Dr. Chu, and Dr. Wu. How do you guys overcome your obstacles? So why don't we start with Peter, please?
Okay, well, as I tried to emphasize in the talk, working with a team is, I always find that to be a much more satisfactory situation than when I'm trying to do something by myself. So as a team, you can, when you get setbacks, at least you have other people to, say, okay, well, let's try that, anyway, you don't get so depressed. You can also have a lot of fun in that. I mean, one of the ways one of the obstacles, obviously, to developing, say, a drug is in an academic setting, is that it just costs so much money to run a clinical trial, and it's really very difficult to do in an academic environment. So is keeping a really good team together over the long term. Academic structures don't lend themselves to that very easily. So that's one of the reasons I've turned to starting companies. I found that if you get a company going, you can keep the team together, and of course then you also have the possibility of raising more money, the kind of money you need to run a clinical trial and perhaps eventually get the drug all the way through. So those have been my tactics. It all started off for me with we invented a device for making liposomes. This was back in about 1985. We called it the extruder. And I gave a talk in New York, and somebody in the audience said, well, I want one of those because I want to be able to make these liposomes as well. And so he called me up, and so I put him on to the machinist who'd made this. And then 2 months later, I got this irate phone call saying, well, this thing doesn't work. And of course, the machinist had no idea what the machine was for. There was no quality testing or anything. So we decided, okay, we'll form a small company and make this. And we had a lot of fun with that. We decided every company needs a mission statement. So our mission statement was free beer every Friday and parties whenever possible. And so nobody in my lab has paid for a beer since then. So that's
wonderful, Peter. I think what you mentioned is obviously teamwork. And when you're faced with resources, questions, or challenges, you just find your own way. I think that's that kind of highlight your entire career, which is that build things that doesn't exist. So it could help you do the things that you need to do, whether start a company or build this extruder. Dr. Wu or Dr. Chu? Dr. Wu, what are some of your obstacles, and how did you overcome?
Yeah, I think all of the discovery maybe take more than 10 years. Yeah. So, but only happy day is only 2 days happy. So I know most periods is fair. So how to overcome? I just jogging, yeah, and mountain climbing, yeah, and change another fear. Don't think about research. Okay, refresh my brain. Then sometimes we get new idea, and then to overcome. Yeah.
That's right. I think we need time to think. We're so busy running around, going to meetings. Sometimes we just need our quiet time to think, whether it's jogging or 3 beers on a Friday. I think that is part of research, is the thinking time, getting yourself back into the game. That's absolutely critical. Dr. Chu?
I'm a physician scientist. I have been very busy in the clinical side. I always find questions from my clinical practice, and I keep my curiosity to drive my research. I find passion on my research. And, yeah, I agree with Professor Gao and Wu. Sometimes you have to relax and reboot, and then you can get the power to keep going on. So, and persistence is very important.
Yes, find that passion. Everybody have a different definition of success. There's no right or wrong answer, but I think once you lose that passion, that curiosity, things start not to be as meaningful. So I think that echoes so well that 50 years Peter has been working on LNP, is that passion, is that curiosity that keeps him going. Thank you, everybody, for that. So there are some other questions. These are more technical questions, so I guess over to you, Peter, which is there are so many negatively charged biomolecules in the tissue, like collagens, elastic fibers, and also glycocalyx. How would that affect liposome uptake? Because they have these unintended interactions along the way.
Yeah, so another good question. This actually leads on to something where we really don't know what's going on, with our lipid nanoparticles. When they're put into serum or into the blood, they immediately get a coating or protein around them, and so they may be positively, negatively charged when you inject them, but by the time that they're within a very short time of being incubated in blood, they have a whole different external character. And I think we're just starting to realize that really plays a huge role in their biological, the effects they have. And an example, obviously, is for uptake into hepatocytes, as I mentioned. These nanoparticles adsorb apolipoprotein E into that glycocalyx or into the surrounding of the protein, the corona, as it's termed, that surrounds the lipid nanoparticle. And that triggers the uptake into hepatocytes via the LDL receptor. So modulating that corona and understanding the composition is highly complicated. I mean, there's thousands of proteins present in the blood, and relating that back to the affinity, the tropism for various tissues, I think is going to be a fascinating, but unraveling that process is going to be is extremely important, and is going to give us new ways of getting these systems to where we want them. But it's very, very early days. One of the big problems from a lipid-based system is that just separating them out once you've incubated them, because they have the same density as LDL, and so the impurities that are there, getting them, finding out which proteins are there is a difficult proposition. Yeah, it's a good question, and we're not there by any means yet.
Yeah, Peter, I think even with things that's working and work beautifully, there's still questions left unanswered. I think instead of looking for answer, whoever posted this question, that could be a wonderful PhD thesis.
Absolutely.
I think that's what research is about, is research. It's not already done. It's you keep on asking new questions, you find out what you don't know, and that presents itself as the next challenge, next question to be answered. Dr. Wu, you have something to add? Yep.
Yeah. Sorry, I can also share some previous my experience about targeting. Yeah, targeting liposome. So previous, as we know, the free drug is very difficult to deliver the high pressure of tumor site. So you can use the liposome. However, we found the targeting liposome, if we conjugate with the peptide or antibody fragment, it can much increase the drug concentration. For example, non-targeting liposome compared with free drug, about 3 to 5-fold in our study, can deliver it to the tumor site. However, if we use the active targeting, then we can trigger endocytosis. So we found more than 10-fold of drug can deliver to the high pressure of tumor site. Yeah. So I think active targeting, is also one of good strategy to deliver a drug or mRNA to a specific tissue or cell. Okay. Thank you.
Yeah, thank you, Dr. Wu. I think you highlight a very important key message, which is there's no just one answer out there. There are many, many answers. I think it requires creativity, innovation. I would encourage especially young scientists, not to pursue the right answer, but to pursue what is possible. I think that is a different way of looking at the world, much more exciting, and you might just learn a few things that along the way. So I have a very good question, and obviously from a student here in Tsinghua University. To Dr. Koulis, what's your interaction with the students in your lab today? And are you interested, any chance, NTHU student can join your lab? So I guess the question is really about that pursuit, that continuing learning. And more importantly, what is your philosophy on the relationship between a mentor and a mentee, or a student and a professor?
It's something I think we all, as academics, we all deal with that. I mean, my attitude is really shaped by being a graduate student myself, and I didn't find that a very pleasant experience. It was you come into a lab and everybody else seems to know what they're doing, and you don't know what you're doing at all. And then, in my case, my supervisor, was. He didn't get tenure, and so after a couple of years in, he disappeared. And so then I had a different guy who didn't know anything about the topic I was working on. But anyway, the overall effect was one of it makes one a bit quite insecure. So I always, with graduate students, I'm very sympathetic to their situation, and I think people, you really do need to treat people pretty gently. Some people do just fine. You suggest, a project, and away they go, and the project actually works. But I have to admit, most of the time the projects I will suggest, well, it doesn't really work all that well, and we have to change things around, and and you have to keep people motivated during that time. Anyway, so I have a very, as I say, a sympathetic attitude, and together we kind of work our way through whatever difficulties there are. And I've had about 40 graduate students now, so it seems to have worked out reasonably well.
And are you interested in taking on some students from National Tsinghua University?
Oh, sure, yeah. No, this is they should mention the Tang Prize in particular, and I do get a lot of people applying for the lab. So now Taiwan has a special meaning to me in terms of new students. So he should say he heard the seminar and wanted to join, and then we'll see where it goes.
I guarantee you won't disappoint. Thank you, Peter. So I think I'm getting just so many questions. Allow me time to sort things out. What are some of the key characteristics in a good scientist? That's a question, and I think it's for everybody on the stage as well. What makes a scientist good?
Is that a question for me, or that's?
Oh, anybody. I mean, any people on the stage, or your good self.
I'll let the people on the stage answer.
Okay, I'll take a stab at it. I think I came across different people in the scientific community along my journey as an undergraduate, as a graduate student, postdoc, and on and on and on. Here I'm sitting on the stage. What I remember the most are people, I don't know if good is the right word, but the most memorable ones that have kind of challenged my view and challenged the way I look at myself, to look at my research topic, or even the world for that matter, are people who are clearly passionate about that topic. It could be a research topic, it could be a social issue, it could be anything. I think when you have passion, when you feel this is something important to you, you would do everything you can to pursue it, whether it's a research question, and you would devote your whole life to it. That's why when Peter started talking, it just pressed upon me that 50 years is a very long time to work on a topic. And I think I admire that, not because he did it, of course, that's fantastic, but because of the passion. He believes in it, that he doesn't know all the answers, and that passion to pursue the next unknown really has my respect. I think if that makes it good, I think, Peter, you're pretty good. So thank you, Peter.
Yeah, pretty lucky as well. So.
Well, luck always plays into it, but you have to go at it all the time. You cannot be lucky all the time. Yeah.
No.
Yeah. So Peter, Dr. Chu, or Dr. Wu, what makes a doctor a good doctor?
Okay, I think the most important thing is you need to feel interesting in one thing or one field. Okay, if you feel interesting, I can work very hard, but you did never feel tired. Yeah. So like someone maybe like the business, some maybe like research. Okay, yeah. But if you can focus, you don't fear the fail. Okay, you never give up. Then you are insistent. Then later on, you will become a professional or an expert. And like the Professor Coolidge can do the lipid and the lipid nanoparticle more than 50 years. Okay, yes, I am very appreciative about Professor Coolidge. Yeah, can insist everything, and finally we will get successful. It's my opinion.
Being a good doctor?
Yes, I want to know what makes a good doctor.
Key point is to put patients in the center always, yeah. As how to become a good physician scientist, I think I always approach the research questions probably more from the applied direction. Yeah, this is a little bit different from the basic science researcher. And of course, the define good or bad. I think not bad. I mean less good. I mean defined by my colleagues in the community or in the public.
Yeah. Peter, anything you'd like to add to this?
Well, there's a couple of things. I mean, one of them is that I've often found that you do experiments. I often learn a heck of a lot from experiments that don't work the way I expected. So where it's not necessarily a good result or a bad result, it's just a different result, and there's something weird going on. I find that those things, they get stored in my brain quite readily, and they can affect, the path I might take subsequently. But it's obviously if the experiment fails, I mean, it's just your hypothesis or whatever was totally wrong. Well, then you're not going to do that again. But as I say, sometimes you get a result, you just say, "Wow, that's interesting," it's not what you expected, but it leads you onto a new path. You can't get too locked in to your own hypothesis. Is the bottom line. Nature can be quite subtle at times, and the way you think about something, you might find that there's really a very different way that the thing was actually happening. So you got to be prepared for that.
Yeah, you just reminded me of something. When I hold group meetings in my own lab, I love it when the student use the word weird data or interesting data, because it means that they're thinking. It means that they are not tuned into their own personal bias. Like unexpected results usually means there's something new to uncover. And when you think about innovation, 创新means something that has never been done before, right? So true innovation comes when you are treading into that unknown, when things look unfamiliar, when things look weird. I think this is kind of that sense of wonder and that sense of curiosity that at least I get a lot out of research. I get that from interacting with students. And I think, again, it comes back to working in groups and working together and that. Okay. So I just want to be mindful, are there people in the audience that don't know or cannot access Slido that have questions? I just want to make sure not everybody is as technical savvy as other people. And I just learned this today, and I had some questions earlier. Anybody, you can just raise your hand. We have microphone. The old-fashioned way also works. Okay, please. We just have 4 minutes.
Okay, hello, Peter. Very nice work, excellent work. I actually worked with Dr. Li Huang 15 years ago to develop several siRNA-based LNP. But after 15 years, still no siRNA-based drug in clinic. So just want to know your opinion. What's the challenge of that in cancer treatment? Especially you have gained big success in siRNA-based drug in genetic disease.
So sorry, the which drug is not in the clinic?
SIR, like RNA LNP, RNA-based drug, LNP, lipid-based siRNA platform in cancer treatment. What do you think?
I see. Yeah, you're asking why they aren't being applied to cancer treatments.
Yes.
Well, I think it's really a matter of well, some people would say it's the low-hanging fruit idea, but what we developed in our with the siRNA are systems that are good for getting to the liver. They don't get to other places very well, and so for going after a distal tumor, well, they don't get there, and so that's certainly one. We've done quite a bit of work trying to with prostate cancer and others, and trying to get to, but it requires getting long circulating systems, and we haven't been very good at that yet. You need to have that different biodistribution so that you're actually hitting the tumor tissue. So I'd say that's the biggest inhibitory factor, is that having an active system that can actually deliver siRNA to a tumor site it hasn't been possible to get a long circulating system that's active up until now, actually. So that's what's holding it back. So this is what needs to be designed: is something that circulates around for 10 hours or so. Actually gets to a tumor tissue and then can transfect the cells in that tissue.
Thank you, Peter. I think in the interest of time, I'm also very aware of the time difference where Vancouver is and where Hsinchu located. I just want to let you know, Peter, that today is the Teacher's Day here in Taiwan. And I know for you, you have to wait until you wake up tomorrow to enjoy Teacher's Day. People in the audience want to wish you a very happy Teacher's Day, and thank you so much for your sharing today. And that and here's the last question. I want to save it for Peter, just for you. As the final question, as a wise and intelligent person, Peter, a serious question: waffles or pancake with chocolate or maple syrup? What's your preference?
Oh, I'm Canadian, maple syrup. So.
All right. There we have the answer. The tea take-home message, maple syrup. Thank you so much, Peter. And I think, allow me to make some final remark. For me, coming to Tsinghua University for the past few months has been a real eye-opening and wonder. I go through my daily schedule, learning new things almost every day. I can say every day. I think it's that sense of wonder and curiosity that Peter shared with us today that makes him who he is and what he did. And I think that's also what we can take away from today's lecture, not just about mRNA delivery, how we have saved COVID, and that's all very critical. Obviously, I'm not belittling any of that. But what makes it possible, the core, is really his passion and curiosity. And I think that is what makes a good scientist great. So thank you so much for joining us online or in person. And Peter, happy Teacher's Day, and wish you all the success in the many, many years ahead. Thank you.
Okay, thank you.